Welcome everyone. My name is Sharon Rounds. I'm the program director for Advance Rhode Island CTR. It's my pleasure today to host the Advance-CTR Clinical Translational Science Research Seminar. Today we have two of our pilot project awardees presenting, and both are presenting their very interesting work that we're really very much looking forward to learning more about. The first is Mollie Ruben. Mollie is an assistant professor in psychology and behavioral science at the University of Rhode Island. And she's going to be talking about race and gender biases in pain assessment beyond the binary patient and beyond the white perceiver. So take it away, Mollie, and thank you.
Thank you so much, Dr. Rounds. And thank you everyone for being here today. I know it is a busy time of year, and I appreciate you taking time out of your days to join me for what I think is a really important topic. I'd also like to acknowledge my co-authors, my graduate student, Addie Weaver, who's worked tirelessly on this project, as well as my mentor on the project, Dr. Lynn Stein, for her support and work on the project as well. And of course, I would like to acknowledge Advance-CTR for their generous support of this project. Disparities in pain care have received much media coverage over the past few years through news articles and podcasts, whereby both personal stories combined with empirical evidence suggest that both women and people of color receive poorer quality pain care and treatment than men and white people. This occurs across the lifespan, pain context, and is not location specific. And I want to point out this New York Times article here at the bottom on unconscious bias in pregnant Black women just came out this week. More specifically, these disparities in pain care that have been documented in the media are not always too far from us here in New England. In fact, this podcast, The Retrievals, documented how Yale's fertility clinic ignored hundreds of women's pain during egg retrievals. It was discovered later these women weren't receiving any pain medication during their procedures, as a nurse was replacing fentanyl with saline. The empirical literature supports many of these more personal accounts. While people of color report higher levels of pain than white people, and work from my lab and others has shown that women report higher levels of pain than men, there is well-documented literature on race and gender biases in pain care. People of color, for example, are 2 to 3 times less likely to receive pain medication as compared to white patients. And women are less likely to receive pain medications and wait longer to receive them than men. So why is pain assessment prone to bias? Well, there is no well-established objective criteria to assess pain, and in medical care, the patient's self-reported pain is considered the gold standard, usually on a pain scale like the one shown here, a numeric rating scale from 0 to 10. The subjectivity of pain leaves room for physicians to question and not believe their patients, especially when they have negative implicit or explicit stereotypes of certain groups or false beliefs about the ability to endure pain, which have all been documented. For example, Burgess and colleagues found implicit stereotypes around who is drug seeking and who can genuinely report pain. Lay people and medical providers have false beliefs about biological differences, such that Black people have thicker skin than white people and thus can endure more pain, or the effect that hardship in life plays on the ability to endure pain. And Lloyd and colleagues has uncovered patterns of questioning authenticity of pain more in people of color than in white people. In my lab, we often induce pain by putting people through a laboratory pain task called the tourniquet procedure. We prompt them to make pain ratings every 30 seconds on a numeric rating scale like the one pictured here, and we video record their experience. We get permission to show their videos in future research. We then recruit new observers to watch short segments of their pain experience and infer pain using the same pain rating scale as the people in the videos used. And we compute a pain assessment bias score by subtracting the observed pain from the actual pain reported, and we look for differences in bias based on the race or gender of the person in pain. For example, across 3 studies, we showed that women of color's pain was the most underestimated by around 3 points, here pictured in pink on a 0 to 10 pain rating scale, followed by men of color in purple, white women in orange, and white men in green. Importantly, only white men's pain was not different from 0, suggesting no bias in pain assessment. However, there are limitations with this past work, even our own past work. Rarely does past work present findings examining biases at the intersection of race and gender. There are well-documented biases in pain assessment for women compared to men and for people of color compared to white people. And our work suggests that women of color experience more bias than men of color that is unique to the intersection of their multiple marginalized identities. This is in line with intersectionality theory, which posits the impact of living at the intersection of multiple minoritized identities is multiplicative rather than additive. Finally, rarely does work examine gender diversity in terms of genders beyond cisgender men or cisgender women. This is a crucial time to examine these unique biases for gender diverse patients, as the number of those who openly identify as transgender, non-binary, or gender non-conforming is growing, while stigma, evidenced by increases in anti-transgender legislation just in the past year, has exponentially grown. These biases likely impact medical care. So looking into biases that exist for gender diverse patients at the intersection of racial diversity is crucial to target interventions in medical education and care. So the purpose of the current research was to examine disparities in pain care in terms of assessment and treatment recommendations for cisgender and transgender racially diverse patients by observers who also vary in race and gender beyond the binary, taking into account important observer explicit biases that have been widely studied in social psychology, but not as widely used in healthcare research. It was hypothesized based on prior literature and theory that people of color, cisgender women, and gender diverse individuals would have their pain undertreated more than white people and cisgender men. Based on intersectionality theory, we hypothesized that women of color and gender diverse people of color would have their pain undertreated significantly more than any other group. We intended to examine observer race and gender as moderators of these biases, but did not posit any directional hypotheses, as past work has shown that pervasive gender and racial biases impact people regardless of their own race and gender. And finally, we hypothesized that observer endorsement of explicit bias toward people of color and transgender people would result in more undertreatment of pain for marginalized groups. While our goal is ultimately to examine these biases in healthcare settings, we first wanted to establish these biases in a layperson sample, as many of the biases that exist in lay people also exist among clinicians. We recruited 819 participants on Prolific, a site that has the ability to recruit a diverse sample of US-based participants. Participants were compensated at an hourly rate of $13 an hour. We purposefully recruited participants by race and gender, so we could have around 200 participants who identified as white cisgender people, white transgender or gender diverse people, cisgender people of color, and transgender or gender diverse people of color. Due to about 8% of our sample not passing data quality checks, our final sample consisted of 754 participants, and here you can see their breakdown of their race and gender. With prior research, as well as ChatGPT, we developed 20 different acute pain vignettes, which described patients seeking care in the emergency department. We ensured vignettes were matched with reliable research assistant ratings on pain severity, the commonality of the injury, how gendered and racialized each scenario was, and age, here focusing all on young adults, though we do think age is an important moderator to consider in future work. Race and gender of the patient varied across 20 vignettes so that every participant saw every combination of race and gender randomly assigned within participant, so this was a fully within subjects design. Said another way, participants saw every combination of race and gender patient in the cells of the table in blue here, and then each patient or cell was randomized within participant to the type of pain or context listed on the right under pain scenario. I'll show you an example of just one of these on the next slide. Participants were given the following instructions. In a few moments, you'll see a real patient's medical record that has been de-identified. The medical record will include information about the patient, including their age, race, and gender. Read this information carefully as you'll be asked questions about the patient's identity. You'll also see a medical record note that describes their injury. Read this information carefully as you will be asked questions about their injury. You'll repeat this process for 20 different real patients. They were then presented with a medical record image. Here's an example of a 25-year-old non-binary Black person who burned their legs with boiling water while cleaning up a catering event. Participants were then presented with the following 4 questions after each medical record. On a scale from 0 to 10, how much pain is the patient experiencing? How much the patient is exaggerating their level of pain? The urgency of this patient's need for pain medication, and how much pain medication should the patient receive. For the purpose of the talk today, we're going to focus only on results for pain medication recommendations. After rating all 20 patient vignettes on these 4 items, participants then completed several explicit prejudice measures to evaluate negative attitudes towards people of color and transgender people. More specifically, participants completed the anti-Asian attitude scale with an example item including Asian Americans are motivated to obtain too much power in our society. The Hispanic Latin attitude scale with an example item, Hispanic and Latin people would be more welcome in the US if they would try harder to learn English and adopt US customs like past immigrant groups have done. The transphobia scale with an example item, I think there is something wrong with a person who says that they are neither a man nor a woman. And the modern racism scale, which is a measure of anti-Black attitudes with an example item, discrimination against Black people is no longer a problem in the US. While we ultimately wanted to examine bias against specific races and genders and at the intersections of these races and genders, for our preliminary results and what we have to present today, we've collapsed observer race and patient race into 2 groups, comparing white observers and white patients to observers of color and patients of color. We similarly collapsed gender diverse observers and patients into a transgender and gender diverse group. Finally, we standardized all measures of explicit prejudice on that previous slide and aggregated them into an observer explicit bias composite. We used a median split to categorize those with high bias from those with low bias. For the analyses we'll be presenting today, we ran a 2 by 3 by 2 by 3 by 2 factorial ANOVA, with the outcome of interest being amount of pain medication recommended. The within subjects variables are underlined here, with the non-underlined variables being the between subjects factors. First, were we able to replicate real-world pain treatment biases and extend to gender diverse individuals? That is, was there a main effect of patient gender such that cis women and transgender and gender diverse, or TGD, people were recommended less pain medication than cis men? We did not find support for any gender biases in pain medication recommendations. Was there a main effect of patient race such that people of color were recommended less pain medication than white people? We also did not find support for racial biases in the hypothesized direction. Patients of color were actually recommended more pain medication than white patients, which is the opposite bias that we see in the previous literature and in clinical care. Was there more undertreatment of pain for patients who held multiple marginalized identities? This intersectional hypothesis was also not supported. There was no significant patient race by patient gender interaction, though white cis men and white cis women were recommended less pain medication than cis men of color and cis women of color. There were no biases among transgender or gender diverse patients by race. Perhaps we didn't find differences in biases of patients because it mattered what the race or gender of the observer was. That is, did observer race or gender, being from a marginalized group versus a non-marginalized group, impact treatment recommendations? Only observer race impacted treatment recommendations such that people of color generally recommended more pain medications than white observers. There were no differences in pain medication by observer gender, nor were there any significant observer interactions. Perhaps patient biases and observer biases were moderated by explicit bias, and that's why neither alone predicted very much. Did explicit bias predict differences in pain treatment recommendations? Yes, explicit bias interacted with observer race, such that bias only impacted white observers in terms of recommended pain medication. That is, white observers with high bias recommended significantly less medication than white observers with low bias or observers of color with high or low bias. There was no difference in recommended pain medication for observers of color depending on their bias. So while a strength included racially and gender diverse patient vignettes and observers or participants, we were not able to replicate many of the real-world biases with these vignettes. So does this mean that biases are getting better? Probably not, as evidenced by this graph to the right in terms of anti-LGBTQ bills introduced in red and passed in blue. These numbers have doubled, if not tripled, since last year, as these data were produced in May of 2023. So it's unlikely that biases against transgender and racially diverse people are getting better in this structural context. In future analyses, we aim to examine racial identity and gender identity disaggregated from people of color and gender diverse groups to examine whether there are biases that we weren't able to detect in the analyses. And while vignettes allow for the careful control and manipulation of patient and context characteristics, they are limited in that participants may become aware of the purpose of the study, which in a fully within design, they may have known we were interested in bias and thus tried to control their responses to appear more socially desirable. Similarly, the participants in our study, prolific workers, could have been knowledgeable about disparities or inequities in care, and unfortunately, we didn't ask about this to be able to control for it or examine it as a potential moderator. Even with these limitations and constraints, we did find that high explicit bias against people of color and transgender individuals among white observers led to lower pain medication recommendations. This suggests we need more targeted assessment and intervention for those who hold more explicit attitudes about patients based on their race and gender. In ongoing and future work, we plan to use real pain expressions to examine these biases by leveraging deepfake technology to understand the impact of race and gender biases while maintaining expressions and all other characteristics. Finally, these data will serve as pilot data in my K award, which is examining medical student racial and gender biases, both explicit, like those measured in this study, and implicit biases, and how they predict communication in a standardized patient interaction with a pain patient. And finally, to end, while we showed today that high bias among white observers led to lower pain medication recommendations, to improve inequities in pain care more broadly, we have to examine the multiple layers of injustice and systems of oppression beyond the intrapersonal and interpersonal processes. Through interdisciplinary research and multi-level interventions that target injustice in policies, structures, institutions, cultures, healthcare, and education, we can decrease biases and disparities in pain care, which is ultimately the goal of my research program. So with that, I will end, and thank you all for being here, and take any questions that you might have in the chat.
So Mollie, it looks like we have a question in the chat about how one of the applications of artificial intelligence is that it can help determine an appropriate pain treatment algorithm to reduce these types of biases. But of course, patients are worried that these behavior algorithms will actually not help. Since changing human behavior is so hard, do you think technology will eventually be able to reduce bias in this space?
I think technology could assist and support, but I think ultimately bias is a human process. And even if people aren't, one of the areas that I study in particular is nonverbal communication. So while ultimately we'll want to reduce treatment disparities, there are these unspoken disparities in care that many patients receive just for being a certain identity. And often that comes through these subtle nonverbal behaviors of spending less time with the patient, being colder and more dominant with the patient, making less eye contact. So while I think that technology has a place in assisting and helping to notify when bias might occur in treatment decision-making, I think that there is a lot of humanness to this that technology can't do, and that we really need to make sure we're educating and training people on these communication skills and how bias might be coming through in their communication.
Yeah, there's another question. Are there specific bias reduction strategies that have been studied and demonstrate effectiveness in reducing caregiver bias?
Yes, social psychology has a lot to say on bias reduction strategies. And I would say there are several that come to mind in particular. One is building empathy and perspective taking of diverse others. And there are specific ways to do this. One of the stereotypes or effects that we have is that when we are not part of a racial or gender diverse group, that we think that people like that are all the same. It's called the outgroup homogeneity effect. And when we treat people like they're part of this group that are all the same, we're not individuating and really listening to what they need and who they are and their specific needs as a patient. So one of the ways that we can get folks to individuate and really see that person as a person rather than part of a group or part of a stereotype is to build that empathy and build that relationship and get to know the person on a deep personal level. So giving more information, asking for more information, building rapport, those are really important components to some of this reducing bias and increasing rapport in clinical interactions. And there's lots of other strategies that have been tested that I could talk probably an hour about, but I'll stop there with just that one.
Fantastic. Thank you very much. And, you know, this is really important work. And as a provider, we're all painfully aware of our biases, that most of which we're probably not aware of. So hopefully a follow-up for this type of work can be education, and including bystander education.
Yeah, I think that's a great note. Yep. Thank you.
Okay. And okay, so we have another question here. We have time for one more. which I'm having trouble reading. So, did you find anything about people of color who demonstrated discrimination against those on the gender spectrum? Literature has shown homophobia within ethnic and racial minorities.
We did not find that observer race impacted bias against our transgender, gender diverse patient vignettes, but that doesn't mean that it doesn't exist. I think I spoke to some of the limitations of vignettes, and I think we really do need to replicate some of this in a more real world context to really understand the effects. I am familiar with the work on homophobia. Here we weren't focused on sexual minority identities, but I would assume if somebody has high levels of homophobia, that they probably also have high levels of transphobia. Those two things often go, are correlated. So it would make sense that that might be the case, but we did not find it in this study.
Okay, great. Well, thank you very much and good luck to you on your K application. Mollie was one of our cohort 4 advanced K scholars, and we're looking forward to hearing more about this important work. So we'll move on to our second presentation. If we could have the first slide. And this will be provided by Dan Spade. Dan is an assistant professor in the pathology and laboratory medicine department here at Brown. And he's going to be discussing his work on sperm RNAs in reproductive health and toxicity. And thank you very much, Dan.
Thank you for the introduction, and thanks for having me. So I will jump right in and say that we are currently funded by an Advance-CTR pilot project, and very grateful for that support. And other than federal funding, we have no conflicts to report. And I want to upfront acknowledge my lab and all of the people who've worked on this project. Really, I'm speaking on behalf of a really large group of people, particularly about when speaking about the clinical project that we'll discuss first. So this has been a collaborative effort between my lab and group at Brown Urology and another group at Women and Infants in the REI division. So a bunch of really great people who do clinical reproductive medicine and have been, willing to work with, with us basic scientists on what I think is a really interesting project. And I should mention also that we've had a lot of statistical help from Dr. Wu and August Guang and a number of other people. So really a big team effort. So I'll talk a little bit about male fertility and semen analysis and sort of our interest in this area. I will cover our human subjects research on sperm RNAs as they pertain to male fertility, and then as time permits, cover some background on another project that we're doing in parallel, which is an experimental project using a rat model with a toxicant, because that's really my background. So for those who are on the chat who do know me, I'm a male reproductive toxicologist, and so the reason why I initially became interested in this topic is because there has been, a lot of discussion in the literature, and I would say in, sort of the non-scientific media as well, about the effects of environmental toxicants, and in particular endocrine disrupting chemicals, on reproductive health. So there are 2 papers here that I'm just sort of pulling figures from that I think have been pretty influential. The one on the left is from a paper that was published in Andrology in 2016. that made the argument that there is an increasing rate of diagnosis of cancers related to, endocrine tissues relative to the rate of diagnosis of all cancers, right? So in other words, testicular cancer, which is the green line on the right, as well as breast cancer on the left and pancreatic cancer on the right, are increasing at a rate that's faster than the diagnosis of all cancers. And so, because this is happening over a period of less than 100 years, the argument is that there's an environmental component that's driving this. On the right is, one of the, I think, most talked about papers in reproductive health research in the last few decades. This was a paper that was published in 2017 and updated in 2023 in Human Reproduction Update. That was based on a meta-analysis of semen analysis publications from 1973 until 2011, which argued that over the course of about 40 years, there is a global decrease in sperm counts that is as much as 50% on average, depending on how they're modeled. And this has been really controversial for a number of reasons. One of the reasons is that it's hard to identify environmental causes of, of reproductive effects, right? Environmental exposures, particularly exposures to endocrine disrupting chemicals, tend to be low-level exposures that happen over long periods of time. But another reason why this is interesting is that really sperm count is not a very good predictor of fertility. And so this was, I think, our segue from reproductive toxicology into questions about, well, how can we measure, what can we measure that might be predictive of fertility that could help us to link environmental exposures to reproductive health in men? And so there's a big question about, semen parameters. I think everybody's familiar with what a semen analysis entails, at least, familiar with the idea of sperm count and sperm motility. But there's a question about how well do semen parameters predict fertility? And so I'd like to show figures from two papers that give a pretty good illustration of the fact that semen parameters predict fertility fairly well up into a point. This is from a paper that was published in 1997, showing, in one study, a really nice calculation of the predictiveness of semen parameters. So receiver operating curve analysis of a fertile population and an infertile population, showing essentially that the area under the ROC curve for most semen parameters is in the 0.6 to 0.7 range. So for comparison, for biomarkers that maybe people are more familiar with, prostate-specific antigen, which is sometimes picked on for not being very predictive, has an AUC of 0.67 at least in one study where it was measured. A really celebrated recent biomarker is KIM1 for kidney injury. So there's an estimate of the ROC AUC for that molecule being 0.93, right? And so, and if you're not familiar with receiver operating curve analysis, basically, 0.5 would be a useless biomarker and a value of 1 is a perfect biomarker. And so, semen parameters are, good, but not perfect. And they have, I highlighted this area on the right of the table that shows that they actually have better negative predictive value than positive predictive value. So if you have very low semen parameters, you can be classified easily as likely to be infertile. But if you have, above reference values, which 95% of people do, it's very hard to determine positively that you are fertile. And this is another really nice analysis showing that, essentially showing that the correlation between the monthly probability of achieving pregnancy and semen parameters increases only up into a point, right? So sperm count or concentration, are correlated with probability of pregnancy up to 55 million per milliliter. Morphology is maybe slightly better. There is a correlation between morphology and fertility up to, depending on the criteria, 19 or about 40% normal. And again, it's better for making negative predictions. So if you have multiple abnormalities per abnormal sperm, you can be classified more likely as being infertile. But so, there's a real question about whether there are better ways to measure fertility. And this has been, I should also mention, one of our collaborators on this is Mark Sigmund, who's been interested in this question for a very long time, because of the fact that it's estimated that about 12% of couples attempting to achieve pregnancy will seek infertility assessment. About 50% of the time, there is a male factor involved if an infertility diagnosis is made. But about 50% of those are idiopathic, right? So there are some cases of infertility that are easy to, I shouldn't say easy, but they can be categorically identified, like azospermia, orchitis, or, infection of the male tract, severely low motility or high abnormalities. But 50% are idiopathic, and the standard of treatment for a lot of patients who have male or female infertility factors is IVF, and IVF is a pretty good treatment. It results in a live birth about half of the time when maternal age is less than 35%. But diagnosis may be, an area that could be improved, and also, identifying etiologies of male factor infertility to determine whether, certain fertility treatments are actually more appropriate, is an area for improvement as well. And I want to just very briefly introduce the idea that sperm RNAs exist and are important, because I don't think that this is something that everybody knows. So I'm highlighting a couple of figures from a paper that was published by Oliver Rando's lab at UMass a couple of years ago. Essentially, this is pretty busy, but showing that immature sperm and mature sperm in the epididymis in mice have different RNAs in them. And those RNAs influence gene expression both before and after zygotic genome activation in embryos. And if you use immature sperm for ICSI in mice, they will have trouble, those embryos are less likely to result in live births after being transferred. Whereas if you use immature sperm for ICSI and at the same time you inject the RNAs from mature sperm, you have a greater likelihood of actually having live births, right? So sperm RNAs exist. They do actually appear to be important indicators of spermatogenic health, and also potentially functionally regulating embryo development, which we think makes them really interesting in terms of paternal exposure. So the first project that we want to talk about is our sort of ongoing effort to compare sperm RNA between fertile and infertile men, using patients that actually present for assessment here at one of the hospitals. And so this first project, which is ongoing, but this predates our pilot project. I'll briefly talk about our sample collection for our pilot project. We are currently having samples sequenced for our pilot project. So nothing brand new, but we'll hopefully have an update soon. So in this initial study, we had 14 proven fertile patients. These are men who meet the clinical definition of proven fertility, which means that they have never used infertility treatment, and they have fathered a child within the last year. They are presenting for vasectomy, presumably because they don't want to have any more children, and so that actually makes them a really good control group for proven fertility. We have an infertility group. These are men that present to Brown Urology for fertility assessment. They are in many cases idiopathic infertile men, although they are a heterogeneous population. We have an established method for purifying RNA from sperm, which is not a simple thing to do. Basically, there is a problem with contamination of semen samples with somatic cells, including epithelial cells and leukocytes. And so we have a well-established method that came from Enrico Bianchi and in Mark Sigmund's group And we're using Illumina sequencing to sequence sperm from these patients. So I just want to briefly mention up front that at the time that these samples were sequenced, looking at semen parameters, the samples that we chose for sequencing were, representative of the whole population, and you can see that, as is pretty typical for comparisons like this, there is really no statistically significant difference between fertile and infertile men in terms of sperm count, which here is density or million per ml. There is a significant difference in motility. So on average, the infertile men have lower motility, which is pretty typical. You would have a hard time actually plucking one of the values from those distributions and determining whether they were fertile or infertile, which means that the predictiveness is still fairly low. And what you see on the bottom is just the samples that we actually sequenced. You can actually see the WHO reference values from 2010. They've recently done away with the reference values, but these are here just sort of to give you the idea that historically the WHO has considered anything above the 5th percentile to be within the reference range for these values. So the vast majority of these patients from both the fertile and infertile populations are within the reference range for semen parameters. One of the things that we have found really interesting is there's actually fairly good separation on the whole transcriptome for these patient groups. So what you can actually see is that there are kind of 3 main clusters in this hierarchical cluster. So in this unsupervised analysis, we capture, 7, proven fertile patients in a sort of outgroup, and another 5 in the sort of second main cluster here. So it's not perfect separation, which you wouldn't expect it to be, but some degree of separation between the fertile and infertile patients on the basis of the whole transcriptome. And then when looking at gene expression on individual genes, what we see is that there were actually over 1,800 differentially expressed genes here, 2/3 of which were, or close to 2/3 of which were downregulated in the infertility patients. And the identity of those genes that are downregulated in the infertility patients are really interesting. So, one of the things that's nice here as a sort of sanity check is that the most enriched group of genes here is, spermatogenesis, which is not an especially novel finding, but something that, that shows us that our data sort of makes sense. But there are novel findings within this group. So not all of the genes that are in the gene ontology biological process for spermatogenesis have well-characterized functions. Some of these genes are basically just here because they're known to be selectively expressed in sperm. And if you look at this plot, basically what it's showing is the downregulated genes in blue, the upregulated genes in red, and the stages of spermatogenesis where they're expressed. So basically, these are all genes that are known to be expressed in male germ cells, mostly in the later stages of spermatogenesis. The majority of them are downregulated, and there are some that are really well characterized. So for instance, protamines are strongly associated with infertility at the protein level. Protamines, of course, are present in sperm because sperm eject most of their histones as they differentiate and package their chromatin in a tighter manner around the protamines. It's been assumed for a long time that incomplete histone replacement is an indicator of immature or incomplete development of spermatozoa. There was a really interesting paper that somebody just sent me the other day, actually, that showed that in a Drosophila mutant that has a mutation that prevents it from ejecting histones properly, that the presence of the histones in the sperm actually causes inappropriate mitotic division of the sperm nucleus upon activation of the maternal genome. And so, there's really good reason to believe that, that there are multiple reasons why histones have to be ejected and protamines have to be present in sperm. But there are also these, without belaboring it too much, there are these transcripts with unknown function that are known to be expressed exclusively in sperm. And so we have a lot of interesting things to follow up on here. This is preliminary, but Han Ji, who is the biostatistician who's been working on this with us, came up with this really interesting summary score analysis in which he did 100 iterations of random splits between the proven fertile and infertility patients, found the 200 most differentially expressed genes in each of those splits, and then tested whether you could actually use those to categorize the men as belonging to the fertile or infertile group. And basically, in this sort of preliminary run, obviously the training set always has an accuracy of 1. The test data have an average accuracy of 95%. Now, granted, I'm not a statistician. I would argue that this is a case of oversampling to some extent, because the model is being tested on some of the same data that was trained on. But we are generating new samples to hopefully test this approach, using totally new data. So the goal of the pilot project, which is ongoing, is actually to expand this analysis to include not just fertile and infertile men, but to include men who are presenting with a female partner who has no known female infertility factors for IVF, so that we can actually compare not just the sperm RNAs that are related to male fertility or male reproductive history, but also the sperm RNAs that are associated with either good or bad outcomes in IVF. So higher rates of blastulation or lower rates of fragmentation, clinical pregnancy, et cetera. And this is in process, but basically the methods have been very similar. We have more samples from the proven fertility group that presented to Brown Urology in the same way. We have IVF participants who presented at Women and Infants Hospital reproductive endocrinology for IVF. I mostly covered the methods, so I won't go too far into the methods again, but the part of this that's in process is the sequencing. We did have to do some additional work to actually standardize the way that the samples are prepared between the 2 sites. So these are prepared in a slightly different way than what we've done previously. And all I can really tell you right now is that with these study participants, we do have lower average semen parameters in the IVF group, so there may be more average difference between the proven fertile and IVF patients than there were in the proven fertile and the general infertility patients. We have sent these for sequencing, and we do have, of the 9 IVF records, we have 4 complete records, including 2 that have gone on to have clinical pregnancies. And so we think we'll generate a really interesting pilot set of data for a future grant proposal, and that's pending. And I only have a few minutes before the chat, so I just want to briefly talk about this second project that I mentioned, which is a project where we've used an animal model to try to experimentally determine what is the influence of environmental toxicants on on these sperm RNAs, and whether we can actually test the effect that they have on embryonic development. And so since I'm running a little bit behind time, I'll just mention that ethylene glycol monomethyl ether is not something that you should be worried about being exposed to, but it is a really excellent model germ cell toxicant. It's very well characterized. We know that it selectively targets primary spermatocytes, which makes it really unique. And we know that 5 weeks after that stage, we can collect sperm that have been damaged by exposure to a short-term exposure to EGME. So what we do is treat these rats for 5 days and then collect sperm after 5 weeks. Our colleague Angela Sturmer published a paper in which she was able to show that this very fine low dose response, where at 75 milligram per kilogram, you get slight reductions in motility, slight evidence of testicular histopathology, but changes in the distribution of the sperm RNAs by class, by category. And what we have found since then, and this is working with August Guang and Yuan Pu, who are biostatisticians who've been working on this, what we found is that there's a really strong association between the level of EGME exposure and the subsequent microRNA exposures. So you can see that there's pretty good clustering by dose, both in the hierarchical clustering and the PCA. And that's on the whole transcriptome, so that's actually pretty interesting. There's an increasing number of significant microRNAs with the increasing dose. And the really interesting part of this is that we have this core group of 12 microRNAs that are significant in all 3 treatment groups and are dose dependent, and they increase. And there are 3 of those that we think are especially important. So these 3 microRNAs are known to be downregulated during normal epididymal development of sperm in the mouse, but upregulated by EGME in our experiments. And so we are, currently devising experiments to test whether these specific microRNAs have an influence on overexpression of these specific microRNAs has an influence on embryo development. And we have plenty of reason to believe that the sort of total effect of these microRNAs should influence development. Looking at the known targets of these microRNAs and doing an enrichment analysis, we see enrichment of categories like regulation of developmental growth, sulfate commitment, morphogenesis of branching structures, and so all really important things for early embryo development. And so we have more work to do on both of these projects, but hopefully, I've convinced you that we have evidence that sperm RNAs differed, at least in this one study of men with different reproductive histories. We have an experimental model that we can use to test how environmental exposures that alter germ cell biology can influence this process. And we have, interesting experiments to do looking at IVF outcomes and looking at embryo development in culture in rats. So with that, I will say thank you, and I'm happy to answer any questions that you have.
Thank you so much, Dan. That's terrific, very interesting work. So I'd like to ask if you'd be willing to speculate. The question I'm sure that you get asked 100 times is, what's been going on in the last 50 years to account for that seminal publication findings?
Yeah, I mean, it's truly a great question, and depending on who you ask, you'll get really different answers. I don't know. I do know that the group that did that meta-analysis are a bunch of really high profile and excellent environmental epidemiologists, and so I would say that nobody really questions the the validity of the meta-analysis. There are certainly people who question whether part of what's driving that trend, so if you ask urologists about this, one of the things urologists will say is that there's not been a measurable change or a measured change, let's say. Nobody has documented that there's been a change in couple fertility that sort of matches that change in sperm count. And so I think the sort of the balancing voices in the room, many of whom are urologists, would say, sperm counts are not great predictors of fertility, and also, it could be that methods have been better. Better standardization of methods over time has led to this sort of downward trend in estimates of sperm count. There are also people who would say that what really needs to exist are longitudinal studies of single populations, and those don't really exist, with one exception in Denmark that doesn't show much of a change. So that's sort of the countering sort of evidence that I would say tempers the fear, I guess, that a lot of people have that everybody's going to be infertile eventually or something like that. I will say that there are lots of, we know of lots of environmental exposures that are toxic to reproduction, and there is a good argument to be made that they should be, regulated in different ways. So right now, the US EPA is considering a proposal to regulate phthalates, for example, which are close to our hearts because we study them, as a class, right? Because they all exert the same kind of toxicity through the same mechanisms, but they're regulated as individual chemicals. And so there's an argument to be made that, all these different things that sort of hit on the same kind of reproductive function are being regulated as individual chemicals, and maybe our total exposure is going up more than we realize it. But there are strong opinions abound. So I don't know, that wasn't a very certain answer, but I guess that's my answer.
And Rich Fryman asks, is it known if sperm motility is dependent upon sperm RNAs?
That's a great question, Rich. So I don't know. I wouldn't say that it's known that motility is dependent on RNAs per se. So I would say that we think that there are two different things that are interesting. So the mRNAs that are present are probably not there to do anything. They're probably just leftovers from spermatogenesis. So there's cytoplasmic exclusion, as you know, but there's a lot of stuff that's left behind in the cytoplasm. And because those cells are transcriptionally silent, they're also not actively degrading RNAs. So there's just a lot of mRNAs that are left behind. And we think of them as sort of a good record of what happened during spermatogenesis. The small RNAs, there's very good evidence that they could be regulating what happens in embryonic development. And so, I think that that's sort of maybe the functional effect. There is evidence that there are some RNAs that are correlated with motility. But I would make the argument that there's no reason to believe that they're sort of regulating motility, at least at the extemporaneously regulating motility. I would say that they sort of fall into that category of being things that are present in sperm that are more mature, and more mature sperm are more likely to be motile. But one thing that we've thought about doing and we haven't done is, and it would be relatively easy to do, is just divide the motile and non-motile sperm from fertile volunteers and compare the RNAs in those populations, right? Which would directly answer your question. And it's known that there are differences in things like DNA fragmentation if you do that motile versus non-motile comparison. So yeah, it could just be a good marker of overall function, in which case it would correlate with motility.
Okay, great. Well, I don't see any other questions. A bunch of compliments for both presenters. These were, very different, but both very thought-provoking and extremely interesting discussions. I do want to point out to the audience that in January, Alan Rothman, who's a professor at the University of Rhode Island, a distinguished clinical and translational research scientist, is going to be discussing his work on risk factors for dengue virus infection and disease. And we have a terrific lineup of speakers plotted out for the rest of the year. So we hope everyone can join us again next month. Recordings will be available, and we would greatly appreciate it if those attendees could please provide us with feedback, which we are greatly interested in and happy to share with our presenters. So thank you, Dan and Mollie. You both gave great talks, and this was really a lot of fun. Bye, everybody.